Animal cloning is the method to produce identical animal from the original source. This can be done by a technique called somatic cell nuclear transfer, SCNT. It is a process which the nucleus of somatic donor cells is transferred to the enucleated recipient oocyte. So the embryo has complete genomic information of the donor cell. This has huge application mainly in livestock production, biomedicine, and genomic conservation. In the past studies, there are methods to determine the genetic source of cloned animal from cloned baby. However, no studies have tried to determine from the level of pre-implantation embryo. So the question of this study was this. This is important because if we find a method and succeeded, we can do these things. Two methods are considered for the study, which are molecular sexing and sequence-based typing. Molecular sexing is a quicker and easier method by detecting the sex chromosome. However, the limitation is that it cannot be applied if the donor cells come from female. Sequence-based typing is more complicated but accurate method, which is by reading a part of DNA sequence of plasmid using Sanger sequencing. In this study, donor cell and recipient oocytes are used as a control, and the sample used to compare with these controls are cloned blastocyst and blastomere. The original donor cell was male Hanu with known sequence of BOLA gene, which is bovine leukocyte genes. After the preparation of the sample, this genetic analysis was applied. The target DNA region used in molecular sexing is specific to Y chromosome. Therefore, if the cloned blastomere has Y chromosome as donor cell, the band of 300 base pair shows up. But if the cloned blastomere does not have Y chromosome as the recipient oocyte, the band does not appear. In sequence-based typing, we choose exon 2 of BOLA DRB3 gene as the target sequence because BOLA DRB3 is the most polymorphic gene in the cow, and exon 2 in this gene is the most diverse among all exons. We use semi-nested PCR for this study to increase the sensitivity because there are limited amount of DNA in the single blastomere or blastocyst. This is a result of molecular sexing. It shows that the cloned blastocyst and blastomere both shows the signal of Y chromosome as same as the male donor cell. However, the recipient oocyte did not. This can tell that the donor cell might have the same characteristic with donor cell. However, this is not enough to conclude that the cloned blastocyst or blastomere belongs to the donor cell. This is the result of sequence-based typing. It shows that the cloned blastocyst and blastomere has identical sequences with positive control. However, there are several differences when we compare with the sequence of recipient oocyte. Therefore, we can conclude that the DNA sequence of single blastomere is same as in cloned blastocyst and donor cell, but different from the recipient oocyte. Overall, both methods can be applied to determine the genetic characteristics, even though there are pros and cons for both methods, and there are some adjustments to be improved. Thank you for your attention.